First of all, I want to thank the organizer of this meeting to invite me to present what we are doing for several years now in the laboratory. And I think the most message I want to transmit to you today is the fact that we have evidence to think that astrocytes are also organized more than in a syncytium way, and they are forming networks. And the purpose of my talk is to give you some arguments to think that there are rules which govern these networks, and I will give some roles which can be attributed to these networks. But first of all, I want just to mention some introduction about the glial cells and about the gap junction. We heard about these two words just before, but I think that we can give some general feature of the organization of the nervous system. Of course, at the center, and this will not be changed by my talk, you have the neurons. They are, of course, the place where information is processed, but there are cells which are actively associated to this process of information, and these are the glial cells. You have here different contributors to the glial cell population. The microglial cell, which are the immune cells of the brain, and the oligodendrocytes, I will not talk about these cells today, which make the myelin around the axon of the neurons. And another partner, which is quite intriguing for the neurons, are the astrocytes. Their name initially come from their star shape that you can imagine here. And what is interesting for this cell population is that they send processes which are closely associated with the synapse, and also, but this could be over astrocyte, not a single astrocyte, they send processes which form end fits around the blood vessels. So these cells are really at the crossroad in the brain. And I just want to point out the fact that the proportion of these cells and the astrocytes versus the neurons is increasing and is up to 1 in the human. And also another point which probably give more interest to look at the property of the astrocytes is that the shape and the size of the astrocyte, this is an astrocyte with GFAP staining of a mouse astrocytes, and you have here with the same scale in astrocytes from the human tissue. And with another way to label all the cell, here you just have the skeleton of the astrocytes. You can see that these cells, again mouse and human, have a very complex and important ramification. So these cells are probably a partner which are important for neurons. And they are important for two reasons. First, this concept of the tripartite synapse is to propose that instead of considering only the presynaptic element with the vesicles and the postsynaptic elements, there are also astrocytic processes which are tightly associated with the synapse. For example, in the hippocampus, more than, I think, 60% of the synapse have associated astrocytic process like this one. And as the astrocytes have receptor, they are not excitable cells, but they have a very elaborate calcium signaling, which could be considered as an excitability property of the astrocytes. They can receive message from the neuron, integrate the message by the calcium signaling, and they can, and this is not represented here, but they can release what we call gliotransmitter, to go rather fast about this notion of the tripartite synapse. Also, the astrocytes, as I told you, are associated to the blood circulation, and Of course, neurons can have some control of the circulation, but also they have tools which are related, again, to calcium signaling, which can either induce dilatation or constriction of the blood vessels. So there is also a control of the astrocytes and a dynamic control of the astrocytes of the blood circulation. Just to end this introduction, we can consider that astrocytes are establishing dialogue with other actors of the brain functions, of course the neurons, the microglial cells, and again the blood vessels. One particularity, and this will be illustrated in my talk, is that these astrocytes express in the brain the highest amount of gap junctions. And this is a typical feature of the astrocytes. What is a gap junction? Just briefly, you probably know that gap junctions are found in most of the tissue of the body. And these gap junctions are in fact an aggregate of intercellular channels, which can be 10 to 1,000, depending on the plaque, the age, and the tissue. At these plaques, the membrane of two cells are getting very narrow, but there is no fusion. There is what is called a gap between the two membranes. This gives the name to this membrane differentiation. And at this level, you have two hexamers made by protein named connexins, which, when the two ring of connexin are face-to-face, make an intercellular channels and allowed direct communication between the cytoplasm. This is the only example of a direct communication between cells mediated by channels. And you will see later, of course, that there is a certain level of selectivity for these exchanges. We know the family which make this channel. They are named the connexins. You have here the organization of this protein in the membrane with 4 transmembrane domain and the N and C terminal, which are cytoplasmic. In the brain, these gap junctions are very frequent, but we know now that these connexins have different function. They have function of communication, and this will be the main subject of my talk. They are the basis of the gap junctional communication. In certain conditions, the hemichannels, which are at the membrane but not at the junctional plaque, can be open and activated and allowed communication between the intracellular and the extracellular medium, like over ionic channels. And there are now other functions which are not directly related to the communication between cells that have been identified in glial cells. This is the case for connexin 43 in the migration of neurons, and also there is some association between connexin and some purinergic receptors. In the connexin, it's a multigenic family of about 20 to 21 members on mouse and the human. And in the brain, at least 11 different connexins have been identified. They have been identified in neurons. They are the morphological and molecular basis of what we call electrical synapses, and they are present as a general feature. They are present especially in the developing brain, and there is less electrical synapse in the adult brain, where chemical transmission, of course, is a major way of communication between neurons. You can see that there are a major connexin. The CX is for connexin. This number, it's the number in kilodalton deduced from the sequence of the protein, but other connexins have been identified. In astrocytes, there are 2 major connexins, connexin 43 connexin 30. Oligodendrocytes have over connexin express, and there is still a discussion about expression of connexin 43 in activated microglia. What you can see from this table is two things. First, there is not only one connexin per cell type, and also the catalog of the connexin present in a defined cell type is different, which make unique the identity part of all these brain cell population. Finally, and I will not talk about that now, there is another family of protein which have been identified, which are named pannexin and are present in neurons. There are some evidence for the expression of pannexin in astrocytes, this is still under discussion. And these pannexin can make hemichannels, but do not form, this is up to now the situation, cannot form gap-junctional channels. So my talk will be related to the expression of these two connexin in the astrocytes and to their role in different brain functions. Just to give you an idea of the level of communication between astrocytes and the other cell type, this is a work that we did a few years ago in the striatum, in the GABAergic striatal neurons. And one way to look at a functional gap junction is to look at what we call decoupling. This is an experiment which was done in a brain slide of the striatum. Of course, we can, with a patch pipette, identify the neurons by injecting currents in the cell. And if the patch pipette contain a dye, which is small enough to go through the gap junction, but is not permeable through the membrane, you will stain the cells that you are recording from. If this cell is what we call coupled to other cell, with time, you can detect the dye in other cell, and this is what happened here. This neuron was patch. The pipette contained biocytin, a small gap junction permeant molecule, and you can see that after 10 minutes, several cells, which are probably neurons based on their size and shape, are also stained. If we do the same experiment, this is in the hippocampus, but in an astrocytes, here again in a slice you can distinguish by the size a neuron and an astrocytes. Once you record it, you can dissociate the firing properties of the neurons and the passive property of the astrocytes. And if this pipette contains biocytin, and for the same time of recording that I showed before for the neurons, you can see that here there are a lot of cells which are stained, about 200 to 300 cells. If we look at the higher magnification, these cells look like what we could think are astrocytes. So what are these cells? The best thing is to do a double staining. Again, this is biocytin, but now it's revealed by a fluorophore. It's in green, and it's a focal plane. And if we make, in the same time, a staining for NEN, which stain neuronal nucleus, you can see that the cells which are green, which are the coupled cell, one cell has been patched and is an astrocyte. The other cells are green, but not red. Even in the pyramidal layer, they are in between the neuron cell bodies. So these cells which are coupled together are not neurons. And if we use either S100 beta or GFAP as marker of astrocytes, you can see that most of the cells are identified as an astrocyte, and we can think that we are facing an astrocytic network. This network is not a general communication between all astrocytes. If we use an EGFP, GFAP mouse, all the green cells are, of course, astrocytes. And if we patch one cell here with, now it's sulforadamine B, another dye which passes the gap junctions, you can see that they are orange cells. But in the area or in the domain of coupling, there are cells which are astrocytes and are not included in the network that we are visualizing with the sulforadamine, indicating that they are excluded from this communicating network, at least at the moment where we are doing the experiment. Another property of this network is to be under the control of active molecules which are present in the brain. This is a work that we did again in the hippocampus. This is a controlled situation. There are even too much cells which are coupled together that we are not counting the cell, we are just measuring the diameter of the decoupling area. And this is the control situation. And you can see here it's an experiment where we have add on the slice and patch another astrocyte with the pipette containing biocytin. This molecule, it's a peptide named endophelin. It inhibits the gap junction channels, and you can see that in this condition, by activating a receptor on the astrocytes, we are able to inhibit this communication and the extent of the network. I will not go in detail, but we did the pharmacology, so we know it's a receptor, we identify the receptor, and what I can mention also is that this regulation of the gap junction mediated network is related to a phosphorylation or a dephosphorylation of connexin 43. We have an antibody which recognizes three forms of connexin 43, a non-phosphorylated form and two isoform phosphorylate. And you can see that after application of this peptide, there is an increase in the dephosphorylated form. Probably there is a signaling pathway which activates a kinase which is involved in this inhibition. Another way to look at this regulation of the gap junction between astrocytes is to do a double patch clamp recordings. Again, this is a slide from the hippocampus. You have one pipette here and one pipette here. We can record simultaneously the two cells, and if we change, the two cells are maintained at the same potential, and if we change the potential in one cell, if they are coupled, we are generating a current through the gap junctions. And the second amplifier, which maintains the potential in the second cell, deliver a current which is equal in sign and opposite in equal in amplitude and opposite in sign to the current which passes through the gap junction. So this is a way to have a measurement at each moment of the level of communication between the two astrocytes. And if we do, in this condition, the ratio between the two currents, the currents in the injected cell and the junctional current, what we call the coupling coefficient, you can see that when we apply the endothelin, there is a reduction of this coupling, indicating that gap junction channels are closing. This effect is reversible, and here we use carbenoxolone, which is a very extensively used inhibitor of gap junction, and we have the same feature. So this indicates that in quite less than 1 minute, you can have the effect here, we can abolish almost completely the communication between cells through gap junction, indicating that there is a regulation which is very potent and rapid of these channels. In the literature, there are other examples. I just give you here a table, but we know that NMDA is, in certain conditions, increasing dicoupling. Kinase seems to have the opposite effect. Endothelin, I mentioned that. And also there are examples where the effect can be dependent on the neuronal activity. I will come back to that later. But there are evidence now that there is a certain level of plasticity of this astrocytic network due to the fact that they are under the control of neurotransmitter or other substance which can be produced. For example, endothelin is produced by endothelial cell in particular. So we were at that point, and then we wanted to go further in the description of the astroglial network. And one picture would give us the idea to go further is this one. In fact, here you have a co-culture of a monolayer of astrocytes on which we have replayed neurons. So the astrocytes are everywhere, you don't see them. The neurons are stained by MAT2, you can see them here. And we have used an antibody against connexin 30. What you have to know is that, and this was initially described by Klaus Willecker lab, in culture, astrocytes express only one connexin, is connexin 43, unless the culture are very old and are, I think, after 11 to 12 weeks of culture. So these astrocytes are 2 weeks old. We put the neurons, and you can see that in this case, we can detect in astrocytes, this is not in neurons, we did a Z analysis with confocal microscopy. We can see expression of connexin 30 in subpopulation of astrocytes. And what intrigued us was the fact that this expression of connexin 30 was closely related to the position of the neurons. It seems to occur when neurons are exactly on the top of the astrocytes. So looking at that, we look for a preparation where we could ask the question how the astrocytic networks are organized in a brain area where there is a strong anatomo-functional organization of the neurons. And for that, we use a well-known system, the system of the barrel cortex, where the whisker of the rodent project with some intermediary step on the somatosensory cortex, and in layer 4, there are what are called barrels, each barrel corresponding to one whisker. So if you stimulate one whisker, one barrel will be light. So there is a strong compartmentalization of the neurons. So the question that we asked was how the communication between astrocytes is organized in this system. And the first thing that we did was to look, in collaboration with the lab of Albert Werker in Lausanne, was to look at the distance between two synapses and between the synapse and the connexin 30. And we measure this distance between two synapses. You have one synapse here, one here, and the distance between a synapse, and here it's for connexin 43, immunoreactive site between two astrocytic processes. And we observe, in fact, that these two modes of communication, synapse for neurons and gap junction for astrocytes, are closer together than between two synapses, telling that probably this proximity will have a functional consequence. And for that, we use brain slice again, and we look at injection within the barrel. And you can see that with this dye, the dye is restricted to one barrel. And when re-ejection is done at a certain distance from the center, there is more coupling going towards the center than going to the external wall of the barrel. And I will not go in detail, but this distance was measured, and we did this type of representation. How this orientation of the coupling, the organization of the coupling between astrocytes occur in this structure. For that, we patch cells and astrocytes. You have here the recording of the astrocytes within a barrel or in between barrels, a region which is named the septum. And you can see here that, in fact, the cells which are in the barrel are quite well coupled together, but the cells which are in between the barrel are not coupled. So these two things, the orientation of the coupling and the fact that there are cells which are not coupled in between the barrel, favor the organization of the communication between astrocytes within a defined barrel, which is parallel to the neuronal organization. We'll not go in more detail, but we looked at the expression of the two connexins, the connexin 43 and 30, and again, in this structure, there is a preferential expression in the barrel compared to other cortical region and the area in between the barrel. We have looked at another structure, and this is a work done by a PhD student in the lab, actually. So these are not published data. We look at another model where there is a strong anatomo-functional organization of the neuron, which is the olfactory glomeruli. You have it there. And the afferents of the olfactory epithelium, which is upper. And we again ask how astrocytic networks are organized in this area. First, we observed that the expression of the connexin was not homogeneous, but there is a defined and a different pattern for connexin 43 and 30 expression. And also, if we perform injection, the Niels coloration allowed to delimitate the glomeruli. And if the injection is done within a glomeruli, there are a lot of cells which are coupled and are part of the glomeruli. They are inside the glomeruli. If the injection is done in between two glomeruli, there are much less cells which are within the glomeruli, and they are rather extraglomeruli, indicating that there is already a compartmentalization of the astrocytic network in this area. The other thing was to look there if the coupling that we described was sensitive to the neuronal activity. And for that, we use TTX to block neuronal activity. And you can see that when we do the injection within the glomeruli, there is a strong TTX sensitivity of the number of coupled cells. But when we do that outside the glomeruli, there is no dependency of the neuronal activity. Also, if we use the knockouts for one connexin, connexin 30 or connexin 43, we observe, in fact, that compared to the control, you have here the inhibitions that I mentioned before. When there is only connexin 43, there is no sensitivity for the injection inside the glomerulus. But when connexin 30 is present, this TTX sensitivity is almost the same as in the wild type, indicating that there is probably a connexin specificity for the sensitivity to the TTX. If we perform the injection outside the glomeruli, there is no sensitivity to the TTX, indicating that there is a region and a connexin specificity of this activity dependence regulation. I have not prepared a slide for that, but we have done also naresis occlusion, which is a long-term way to block the neuronal activity on one side of the olfactory bulb. And in this case, we find exactly the same result. The fact that inside the glomeruli, we have a reduction of the coupling compared to the contralateral side. And also this effect is only observed in the connexin 43 knockout mouse. So short-term and long-term dependency to neuronal activity. And as a first conclusion of this part, which describe properties of this network organization, I just want to mention again that when we inject one astrocyte, most of the cells are identified as astrocytes. So we think we are facing an astrocytic network. This coupling can be, and this network can be regulated by endogenous compound, including neurotransmitter. This, in certain area, and I will come back to that for the hippocampus, in certain area there is an activity dependent of this network. And as I show you for the barrel cortex or the olfactory bulb, in area of the brain where there is a strong anatomo-functional organization, and the network are also restricted to some extent in this area and are superimposed to the neuronal circuits. Okay, what could be the role of this network? This was a question we wanted to address for many years, and what we were lacking was tool to do that. What we know for the gap junction is that they are permeable to ions. It's what I mentioned here. Of course, there is what we call an electrical coupling, but they are also permeable to small molecules, up to 1 kilodalton, 1.2 kilodalton. This depends on the nature of the connexin which are making these channels. In astrocytes, there are quite information about this permeability. Most of them are coming from culture cells, and now we start to have some information about permeability of gap junction in brain slice at least. But these channels, and there they are mainly made of connexin 43, they are permeable to glucose, to lactate, to glutamate and glutamine, IP3, ATP, glutathione, and cyclic AMP, etc. So there is really a basis to have a chemical or a biochemical dialogue between astrocytes through these gap junctions. And one of our purpose was to try to found argument to say that in addition to consider the neuroglial interaction only at a single cell level, like the tripartite synapse, due to their possibility to exchange many signaling molecules, the interaction between astrocyte and neurons should be also considered as interaction between networks of neurons and network of astrocytes. And of course, there are, in the literature, quite a lot of examples where the gap junction can be involved in astrocytic function. They are named here, but my purpose here is to go in more detail about the fact that gap junction in astrocytes can be involved in the trafficking of energetic compound. This is the next part of my talk. And this starts quite a long time when we wrote a review, and we show a finding done in Canada by Giaume, where using the connexin 43 antibody, he shows that in the cortex, at the level of the blood vessel, there was a very strong immunoreactivity for the astrocytic connexin 43. And by EM, he really shows that this was in between the end feet of the astrocytes and not between the endothelial cells. So there is a large possibility of communication between end feet in an area which is very close to the blood circulation. In a more recent work, we did the same type of experiment in the hippocampus for the two connexin, and again, you can see this large expression of connexin at the end feet, the contact of the GFAP process around the blood vessel of connexin 43 and connexin 30. This is between the end feet of the astrocyte. If we stain the endothelial cell with PCAM, there is no overlap for connexin 43, and this indicates with the GFAP staining that this expression is between the astrocytic end feet. If in a slice from the hippocampus, we patch one perivascular astrocyte, in the first minute after the recording, the dye preferentially is diffusing along the blood vessel, indicating probably that at that area of contact between the end feet, there is a high level of gap junction communication. If we wait more, of course, the dye coupling is increased, and if we block the gap junction by carbenoxolone, we just have the recorded astrocytes. So, looking at the position of this gap junction and the fact that the blood circulation is very close, you know, of course, that the source of energy for the brain is the circulation, which bring the glucose to the brain. And and I will show that in a schema in a few minutes, the source are the neurons. And there is a pathway which includes the astrocytes. So many years ago now, we first wanted to know if gap junctions were permeable to glucose, and at that time we use radiolabeled glucose. And here what you have, it's a cover slip with a monolayer of astrocytes. We load the cell by scraping, and of course this is a very weak magnification. And you can see that this diffusion that you see here, it's reduced in the presence of endothelin, or when we use a cell line which is devoid of connexin, or at least with very few connexins. So this indicates that glucose can go through gap junction, and this diffusion can be blocked by closing the gap junction. But we wait quite a long time to have tools to investigate this question in brain slice, and there were a few years ago the finding of two molecules, which are the glucose plus a fluorophore. This allowed us to follow the diffusion of glucose within the network of the hippocampal slice. And you can see here that using this fluorescent glucose derivative, we can see a network of communicating cells. It is blocked by the gap junction blocker, so it's gap junction mediated. If we use the knockout that I mentioned before, connexin 30 knockout, connexin 43 knockout, you see that there is a significant reduction of the coupling. In the double knockout, there is no communication at all. And of course, we did also injection of passive dye, which can pass these gap junctions. If we do the same experiment in neurons which are not coupled or weakly coupled, you can see that the same time of recording maintain the dye V2 and BGG in the pyramidal cell or in the interneurons. And if we look at the neuron properties during these recordings, we have no change, indicating that we are working in conditions where we don't modify neuronal activity and neuronal properties. So using this dye, we look at the effect of changing the activity of the neurons. This is a control activity in a pyramidal cell with some spike. TTX, of course, block the activity. And using zero magnesium and picrotoxin, we can induce epileptic-like discharge. And if we look at the extent, of course, in same condition of recording, you can see that compared to the control, when the activity is reduced, we have a reduced diffusion of this glucose. And when the activity is increased, we have an increase in the size of the network. We have done some pharmacological analysis of this, and what I wanted to point out is that when we use passive dye, which is a way to have an assessment of the level of communication, we did not modify the gap junction permeability. This indicates that probably what is happening here, and this is particular to the hippocampus compared to the olfactory bulbs that I described before, there is no activity dependence of the gap junction in this structure, and this change in the size is due to the fact that there is more or less neuronal demand of glucose. To give more argument to this statement, what we did is to take advantage of the architecture of the hippocampus, and we did injection in an area. Here you have the pyramidal layer. We did the injection on this side of the pyramidal layer, where only 20% of the afferents of the Schaeffer collateral are making synapse here. Most of the synapses are here. So when we do the injection close to the pyramidal layer, you can see that the network that we are visualizing is restricted to this area. If in the same time we make a stimulation of this afferents, we increase mostly the synaptic activity on this side of the pyramidal layer, and you can see that at that time we change the shape of the network, and the glucose is now observed in astrocytes where the synaptic activity is mostly increased. And what is important also is that the number of coupled cells is not modified, it's only the shape. This indicates that probably when there is a source of active neuron and synapses, there is a more requirement of metabolites. So we describe this metabolic network in the hippocampus. What can be its role? And this is a model which was proposed by Pierre Magistretti quite a long time ago for the delivery of energy substrate to neurons. Again, the source of energy is in the blood, and the most active site for consumption of energy are the synapses. So, of course, there is a pathway which just takes into account the extracellular space because the neurons have glucose transporter. But due to the fact that astrocytes surround the blood vessels and have glucose transporter, they are also involved in the delivery of energy to the neurons. And this is done, I will not go in detail, to the fact that the glutamate takes some ATP, which require uptake of glucose and transformation in lactate And it was hypothesized that it was not glucose but lactate which was delivered to the neurons. Again, you can see here that only one astrocyte is figured. So what we did is to start with the hypothesis that the fact that these gap junction channels are permeable to glucose, in fact, this schema should also include the fact that some of the glucose or derivative of glucose could be exchanged between astrocytes, and that the metabolic networks that I showed before could be involved in this delivery of energy substrate to neurons. For this, we took advantage of animals which were provided by Klaus Willecke. In the wild type, when we do an injection, of course, with here sulforodamine, we have communication between the cells. And in the knockout, when we do the same experiment, the dye is restricted to the astrocytic network. So we have a preparation where we can compare a situation with an open network and a closed network. Okay, we take again advantage of the architecture of these astrocytic, of the hippocampus, sorry, and we look at stimulation of the Schaeffer collaterals. We measure the extracellular field, which is an index of synaptic activity, and we patch one astrocytes in different conditions. This is an experiment which was first done in the optic nerve by Bruce Ranson. And of course, when you want to maintain an acute brain slice alive for 6 hours, okay, you have to put glucose outside to maintain the slide alive. So if you take out the external glucose, what happened to the field? It's after a certain moment, which is due to the stock of glucose which are in the glycogen of the astrocytes. After that, there is a reduction of the amplitude of this response because the synaptic activity cannot be maintained due to the lack of glucose. If in the same time, in the pipette where we record from the astrocytes, we put glucose. It's not the fluorescent glucose now, it's just glucose. The glucose from one astrocyte will diffuse in all the network, and we can maintain the activity. This is shown here. If we do the same experiment with a knockout for connexin, there is no open network. And in this case, when we have zero glucose, in control condition or with glucose in the pipette, we don't maintain the activity. This indicates that the opening of the network allowed to maintain neuronal activity. And if we do the same thing with lactate delivered to the astrocytes, we maintain the activity. This indicates that, in fact, it's not glucose which is given to the neurons, but lactate. For just to be more close to the topic of this day, I show also a situation where instead of stimulating the afferents, we use zero magnesium and picotoxin. We have epileptic-like discharge. When we withdraw the glucose, there is no more activity. This is reversible. And if we give glucose to the astrocyte network, in this case, we can maintain to a certain extent the epileptic activity, indicating that the network can be involved in delivering energetic compound. So this is one role of the astrocytic network, and I just make this brief conclusion that gap junction-mediated network provide a pathway for the trafficking of energetic compound. This trafficking is depending on neuronal activity, and this trafficking help neurons to maintain their activity. Of course, I want to say a few words about the pathological situation, and just to give you the idea that the literature so far is not very homogeneous. There are some contradictory data, probably due to many things, and also for the models which are used, for example, for epilepsy, where both increase and decrease of coupling have been described. And one thing also is that there are mostly description of change in the astrocytic connexin rather than functional study, and I think that this is lacking to the field. And I just want to mention that the group of Christian Steinhauser here has worked with the double knockouts that I have mentioned, and they arrived to the conclusion that gap junctions are involved, but not exclusively involved in the control of the potassium homeostasis. But I just wanted to mention this thing, and probably I will not have time to do that. I just mentioned that we are working on a model of Alzheimer's disease, a double transgenic mouse with amyloid plaques. And at the level of these amyloid plaques, there are reactive astrocytes. These reactive astrocytes show some spot of immunoreactivity for connexin 43 or connexin 30. And in most of the case, we see an increase in the expression of this connexin. And this could be a way to explain the fact that the group of Helmut Kettenmann has shown that coupling is increased in animals where there is an overexpression of the precursor of the beta amyloid. And of course, I have no time to talk about intercellular calcium wave, but in the same models that I mentioned in our hand, they show that the calcium wave are also involved. So this could be another function of the astrocytic connexin in a pathological situation. And these are the people who have contributed to this work. Laurent and William have done the double patch clamp recording. Lisa, it's a PhD student, worked on the olfactory ball. Vanessa on the sensory work. Natalie was first a PhD student, but now she's a junior scientist, and she was responsible of the work of the glucose that I presented. And again, the mouse were coming from Bonn, from Klaus. I thank you.